Hello everyone, welcome to the class Introduction to Bioinformatics. This is Yue. In the following 3 weeks, we'll talk about next-generation sequencing, including technical review, read mapping, variant calling, and RNA-seq abundance estimation. Before I start today's lecture, I want to acknowledge the BISA team for their great help in preparing these lectures and hands-on labs. BISA is a shared resource in bioinformatics at OSU. Gutin is our senior advisor and Majay is our technical director. The team members have robust expertise in high-throughput, high-dimensional data analysis. If you have any questions regarding sequencing data analysis or experimental design, please feel free to contact BISA. This lecture contains 4 parts: sequencing technologies, the quality assessment, mapping algorithm, and the mapping representation, that is the mapping data format. Let's start with the sequencing technology. Next-generation sequencing has been extensively used in studying genetics and genomics. These two study fields sound alike, but there are some important differences between them. Genetics is the study of individual genes. The keyword is individual genes, mutations within those genes, and their inheritance pattern in order to understand the specific traits. It dated back to scientist Gregor Mendel in middle 1800s. And in contrast, genomics expands upon classical genetics and considers aspects of the entire genome. Here the keyword is the entire genome. And the genomics term first raised by Jason laboratory scientist Tom Roderick in 1986. So genomics is a relative much newer field and become possible only in the last couple of decades, thanks to the advances in DNA sequencing and computational biology. Talking about the DNA sequencing, it's the process of determining the order of nucleotide bases, ACGT, in DNA. It has 3 generations. The first generation is Sanger sequencing. It's very accurate, still the golden standard so far. It has a read length about 400 to 900 base pair, and the drawback is it's low throughput and expensive. The second generation sequencing includes a number of different modern sequencing technologies, from Roche 454, Illumina, Roche Solid, Ion Torrent, and recently BGI sequencing. It has very high accuracy, high throughput, and cost efficiency. The biggest disadvantage of second generation sequencing is its relatively short read length, from 50 base pair to 250 base pair. This drawback, the short read length, is already overcome in some way by third generation sequencing technology, which include PacBio and Oxford Nanopore. The features of third generation sequencing is it has very long read length. In average, it's between 10,000 base pair to 23,000 base pair. And for Oxford Nanopore sequencing, it can even reach the maximum over 2 million base pair. So it can provide very useful information in genomics complex region, like the repetitive region. And the drawback for this third generation sequencing is still have relatively high error rate, the low accuracy, and the throughput and the cost is still relatively worse comparing to the second generation sequencing. Here I'm showing you the world map for high throughput sequencers. I got this map from this website maintained by James, and James also has a very interesting blog, has comments in the recent NGS industry and a summary of all kinds of different sequencing technologies. So look at this world map. The red color indicates Illumina, orange is LEFTAG Ion Torrent. So these are the 2 major kinds of next generation sequencing technology. And the yellow PacBio, Green, Oxford Nanopore are the newcomers, the third-generation sequencing technology. They are still have a relatively smaller amount compared to second-generation sequencing technology. So the most of the sequencers are located in United States, Europe, and China. Actually, I think this map is kind of outdated because as I know, the BJI in Shenzhen has, even though they have a lot of Illumina sequencers, but nowadays they started using their own sequencer, BJI sequencers. And also, Chinese scientists have recently sequenced the whole genome of the coronavirus. From the first patient sample to the final genome sequencing deposited to gene bank, it only take about 1 week. So this is also a application, an example, see how the next generation sequencing technology contributes to the outbreak surveillance. Okay, anyway, the main message delivered by this map is that the Illumina sequencing technology is still dominant next generation sequencing technology, and that is what we're gonna talk about in detail in the next few slides. So the basic principle of Illumina sequencing technology is the reversibly terminated fluorescently labeled DNTPs for synthesis and sequencing. It sounds a bit complicated, but I will explain it step by step in next few slides. And I also recommend you to watch these 2 videos for better visualization. Also, for all the previous sequencing technologies, including third-generation sequencing technology, you can just type in the name in YouTube, and there will be very good movie cartoons show up and can explain the detailed technology. So the first step in Illumina sequencing technology is to prepare flow cell. In the top left picture, we see this is a chip of the size of a glass slide. Inside this chip, there are 8 channels or 8 lanes. The inner surface of this lanes is chemically modified by 2 kinds of DNA primers, the purple one and the blue one. These 2 primer sequences are complementary to the adapter sequence of the DNA library to be sequenced next. And these 2 primers are covalently linked to flow cell. The reason for the covalent bonding is because later on there will be a lot of fluid to flow through this flow cell, and only those covalently linked DNAs will not be washed away. This is the flow cell primer cluster generation. The second step is to prepare library. First, genomic DNA is fragmented with ultrasound or enzyme digestion. Second, we ligate adapters, the purple and the pink color, onto the fragmented DNA, the gray color insert sequences or fragments. And then we denature the double-stranded DNA into single-stranded DNA by heating to high temperature. The third step is to do the bridge PCR amplification. This is a key step for massively parallel sequencing and for high throughput sequencing. So I will explain it slowly. In this process, the library is first added to the flow cell, and here the green chain is the primer and the black stuff is substrate, is the flow cell surface. This blue chain is the DNA fragment with adapter, and because the sequence of the adapter is complementary to the primer on the chip, so the complementary hybridization occur. And after the hybridization, we add the DNTP and enzyme, the polymerase, onto the flow cell. The polymerase start with the green primer and synthesize a whole new DNA strand along the template. This new DNA strand is completely complementary to the original sequence. Next step, we add a sodium hydroxide, the basic solution, and the DNA double strand was melted in the presence of sodium hydroxide solution and was washed away by the liquid flow. The original template strand, which was not covalently connected to the chip, was washed away, and the newly synthesized chain covalently connected to the chip was retained. Then we add a neutralizing liquid to the flow cell to neutralize the alkali solution. When the entire environment becomes neutral, the other end of the DNA strand will undergo complementary hybridization with another primer on the flow cell, that is the red color or orange color primer on the flow cell. So next step, we can add the DNTP and the polymerase again, and the polymerase extends the second primer to synthesize a new strand. Then add another base solution to dissolve the two chains, leave only one chain. Then add a neutralizing solution to hybridize the DNA with the new primer, and add the DNTP and enzyme again. Repeat the whole process again, and the number of DNA strands will increase exponentially. This is the bridge PCR amplification. After bridge PCR amplification, the reverse strands are cleaved at the primer 1 position and washed off, leaving only the forward strand for sequencing, or the forward strands are cleaved at the primer 2 location and washed away, leaving only the reverse strand for sequencing. The fifth step is sequencing by synthesis, and the sixth step is imaging. In nucleotide addition step, fluorophore labeled terminally block the nucleotide or the DNTP hybridized to the complementary base. Here I'm showing you the chemical structure of DNTP. It's fluorophore labeled, and its 3' end is blocked. This blockage is reversible. That's why we call it a reversibly terminated DNTP. And because the 3' end is blocked, it can only extend one base per cycle. After synthesis, we rinse off excess amount of DNTP and enzyme with water, and in the imaging step, the flow cell was put under the microscope and go for laser scanning to determine which base according to the emitted fluorescent color. And after imaging, chemical reagents are added to cut off the N3 group, the blockaged group, and the labeled fluorescent group. After cutting off, the hydroxyl group at the 3' end is exposed. And next, we go back to the nucleotide addition step, then imaging step, then cleavage step. This cycle is repeated hundreds of times, and you can read out the sequence of hundreds of bases. The number of cycles is related or is roughly same as the read length you will get. This slide shows the overall workflow from bridge amplification to sequencing by synthesis to imaging and the final reads base calling. So here I only show 2 example reads with read length 6. The read generated according to the color from a series of the imaging. From the first one, the green color corresponding to the base C, and the blue corresponding to A, and the red T, green C, yellow G, and the red T. So usually we will get reads with length from 75 base pair to even 300 base pair according to the Illumina sequencing technology. So this is the length after removing adapters and the index. So talking about the indexed, what is index? So remember the adapter 1 and the adapter 2 in the sample preparation step. For one adapter at both adapters, we can add a specific sequence, the orange color. This particular sequence is called index, or some people call it barcode. And reads with the same index usually belong to the same sample. The purpose of doing that is because the high throughput of Illumina sequencing, usually it can generate hundreds of millions of reads in one run, but the one sample usually does not need so many reads. So scientists come out of a way to make some marks on the library adapters using those indexed, so they can mix many samples, hundreds or dozens of samples can be mixed in one run. And then after the run, the scientists can divide those reads according to their index. So usually the pre-designed index is 6 to 8 nucleotide length. And here in this website, it has several Python scripts to design index or split the FASTQ file, sequence files by index. You can take a look. And another core technology of Illumina is pair-end sequencing. Pair-end sequencing means that a DNA strand can be read from positive direction and from negative direction. Let's review our step 4 in previous slide, page 9. Depends on which primer you cut. You cut primer 1, then the DNA fragment can read from positive direction. If you cut primer 2, then wash away primer 1, the one strand, then the fragment can be read in negative direction. So this is the one fragment. Depends on the read length and the fragment length. The insert size is a fragment cut the read 1 and the read 2. The middle part, it can be positive, and sometimes it can be negative. If you read through the fragment, then the insert size can be negative. So this is very useful. The pair and read can provide very useful information to map the reads over complex region, genomic region, like repetitive regions or insertion deletion regions. So it's very useful for applications such as de novo genome assembly or large structure variation. On the other hand, for counting or profiling the gene expression or differential gene expression analysis, usually single-end read is typically sufficient. Let's look at the Illumina sequencing platforms. iSeq, MiniSeq, and MySeq are benchtop sequencers good for laboratory with small to medium size. It can generate output from 1 GB to 15 GB, and the price is less than 100K. So the runtime is typically less than 2.5 days, and the maximum read length varies between 150 base pair to 300 base pair. And the number of reads can be generated per run from 4 million to 25 million. So this is good enough for small laboratories. And for production line or for sequencing course like the one we have in OSU in the nationwide Children's Hospital sequencing core, they have the NextSeq, HiSeq, and the NovaSeq sequencers. So HiSeq 4000 is a very good platform. Unfortunately, Illumina already discontinued the production of the HiSeq series and is replaced by NextSeq and the NovaSeq nowadays. NovaSeq can have up to 6 terabyte output per run and 20 billion reads per run. So let's look at the NovaSeq 6000. It has 2 flow cells, and for each flow cell, depending on different flow cell type, it can have tens of gigabyte output or up to 3 terabyte output per flow cell. And depends on different applications. For one run using dual flow cell, it can have 48 whole human genomes one run, or 500 whole exons, or 400 RNA-seq. That's very impressive. That's based on 30x genome coverage, or 100x exon coverage, or 50 million reads for RNA-seq. Usually we don't need 50 million reads for RNA-seq. And Remember, for the first human genome project, it took us more than $3 billion and over 10 years to finish one human genome. And nowadays, we can already finish 48 human genome within 2 days. That's a very big improvement in DNA sequencing technology.